Okay, good morning, everyone. Thanks very much for having me here today. It's really quite a pleasure, and it's always fun to talk about food safety right before lunch, right? So the question that I would like to frame in the next few minutes is really looking at how does soil contribute to emergence or reemergence of protozoal and viral pathogens on fresh produce? A lot of the organisms we're going to talk about over the next few minutes are really more waterborne, and so we think more often of how are they found in surface waters, what are questions around reclaimed waters, but how does that now contribute to what's happening in the soil? And as we've heard earlier this morning, discussions on climate change and what happens when we don't get those deep freezes that we used to get. Oocysts and viruses can certainly persist in water for long periods of time and can persist in soil for long periods of time as well. Some really interesting things to think about in terms of parasitic disease in particular, and I don't mean to be avoiding the helminths, but we're really going to focus on protozoa for a few minutes now when we think about changes in the United States. The Centers for Disease Control and Prevention has noted a 45% increase in the number of cases with cryptosporidiosis over the past couple of years. 60 million Americans are believed to be infected with Toxoplasma gondii, and that's noted as one of these neglected parasitic infections. CDC has added 5 of those, and toxo is one of them. Toxoplasma also is the third leading cause of death associated with foodborne illness. So these are pretty important agents we're talking about. And if you've been following the news, you've probably seen cyclosporiasis in the news with outbreaks associated with Cyclospora cayetanensis. So I brought up my cheat sheet so I'd get the numbers right. 2,299 cases in 2018, 1,065 in 2017, and 384 in 2016. But we've seen these kinds of general trends of increases in terms of cases of cyclospora with imported produce, but also with domestically grown produce. And so that really makes us come back to how are we growing our produce, what's happening with water, what's happening with soil, how do we understand that a little bit better? And so I really have these kind of 3 things to be thinking about: detection, monitoring, and control. And certainly with viruses, we have to include those there. We think about hepatitis A and norovirus really being transmitted by humans, and we have 21 million cases of norovirus each year in the United States. The CDC estimates we will all experience norovirus sometime in our life, 5 times actually in our lifetime, so quite a bit there. And we've seen hepatitis A also increase, 4,000 cases in 2016 in the United States. Not all foodborne, but something to think about, that it's there in the environment, and how do we get rid of it? How do we control it? How do we monitor for these agents? So if you're not quite familiar with the organisms I'm talking about, I thought I'd give us a little bit of a baseline here. The center image is the life cycle of Cryptosporidium parvum. So that little top point there is an oocyst, and that's ingested. And then once ingested, these oocysts go through rounds of asexual and sexual reproduction in the human gut, and then the oocysts are shed in feces. And so they can be transmitted then by manures or biosolids that may be land applied, or septic system breaks that happen, such as Michelle just mentioned. And if you look to the left, there are 2 oocysts that are there. The top one is Cryptosporidium parvum. That one's a zoonotic agent. The lower one is Cyclospora cayetanensis. They look fairly similar, but they're a little bit different in terms of how they're shed, whether they're infectious right away or they need some time in the environment to become infectious, and then how they propagate and how they survive. And that membrane there on the outside is really tough. And so we think biologically, what's happening in our soils? These things bind to colloids, they stick to them, and so the differences in the soils are really important when we think about how these might be transmitted in the environment. One thing that's happening with cyclospora is that we learn a lot in terms of how these agents are transmitted and how they survive from epidemiological studies. And I just mentioned these years of very large outbreaks. One thing that a lot of scientists right now are asking is the biology we know about cyclospora different than what's really happening? And is that why we had over 2,000 cases in the United States this year? You know, we don't know much about it because we think it's only transmitted by humans, and so we have limited abilities to study it. So So on the right-hand side, there's 3 viruses there. Adenovirus is a DNA virus, and it looks really different. It has those spikes. That's why I thought I'd add it there, because when we think about transmission in the soil, it behaves very differently. Those spikes really bind differently to the colloids, and it moves differently. And then norovirus and hepatitis A are also very different. Even though they're so small, 38 or 30 nanometers in size, they are attaching to these colloids in different ways. And so despite so much improvement in terms of scientific questions related to pre-harvest bacterial transmission, we still have these huge data gaps when it comes to viruses and protozoa. How do we study them? What do we use to monitor those? Do we use the zoonotic agents? Do we try to use surrogates? So that first picture there shows human norovirus and 2 very popular surrogates. My laboratory uses both Tulane virus and murine norovirus, quite interchangeably with human norovirus. We've actually done some hydroponic work with Tulane virus, but thinking about being able to grow liters and liters of these organisms and spray them onto soil in a field trial is a pretty tedious thing. So how do we go about trying to develop these studies, these multidisciplinary studies, to be able to understand how these move in the environment? Do we use molecular detection? That certainly has its limitations. What kind of models do we use? Do we get our data together and then go to the risk modelers? We really need to use all of this. I think when it comes to what answer is best for these questions, I'm a proponent of using them all and then trying to feed all that information together. And then getting control strategies is pretty tricky. We don't understand quite how they survive. If we again look at those 2 oocysts down there on that lower picture, cryptosporidium is really small. It's about 4 microns in size. It's less than half the size of cyclospora, but they behave really differently. So because we can produce cryptosporidium in a calf or even in mice models, do we refer to those? And how do we interpret that data? So it's pretty complicated questions. A few more images for you, but I think if we're going to be looking towards these multidisciplinary approaches, looking for funding together, that I have these sort of 4 themes that I'd like to offer. One is, how do we look at presence in the environment? How do we think about how the protozoa and viruses interact with soil in particular, and so many different types of soil that are available? Also this discussion on these high nutrient amounts of soil. Pat showed some really interesting data on what affects the bacteria. Well, how does that affect protozoa and viruses? How do we think about exposure that's happening? I'm not so certain that we have thousands of cases of cyclosporiasis because we've had 1 or 2 infected workers working on a produce field, for example. And when we're involved in litigation of cases and understanding of these outbreaks, I end up with a lot more questions than we get answers for, because I'm really not quite sure that the biology that we understand is really how we're getting transmission. And so if it's not, how can we test that? How can we better understand it? It's going to take a lot of different fields of science for sure. And then how do we transfer that to crops? So that lower pie graph there is actually some research that we're doing with the FDA, where we grew cucumbers in the presence of a generic E. coli. And over the harvest period, we sampled 160 cucumbers, and we were able to detect E. coli and naturally occurring salmonella from the poultry litter that we used to amend that soil onto those cucumbers. It's really difficult to try to do that with oocysts or with viruses. Right? The production scale is just so different, and the detection and the loss that comes. So how do we understand those different things? The picture there on the left is actually romaine lettuce, where viruses attached to biosolids were able to get into the stoma of the leaf. And they're actually transmitted more easily when they're attached to these colloids from soil and from manure. And so if we know that, how do we model that behavior when we're thinking about transfer onto the crops? So many different questions there, but I'm really excited about this audience and about trying to better understand all of these aspects of food safety, and hopefully that gave you a little bit of insight into viruses and protozoa so we don't leave them out of the conversation. Thank you.